This Week in Virology, the podcast about viruses, the kind that make you sick.
Hello, Coyotes. Is that the best you can do? Let's try it again. Hello, Coyotes. All right. It's time for This Week in Virology. This is episode 338, and today is May 21st, 2015. Hey, everybody. I'm Vincent and you are listening to TWiV, the podcast all about viruses. Today we are coming to you from California. We are in San Bernardino at the California State University, and joining me today, all the way from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How you doing, Kathy?
Doing fine. I'm enjoying it here.
What's the weather like here?
Well, it's 61 degrees outside. It feels like 61. Humidity is 60%, and for those of you that want it in centigrade or Celsius, that's 16. There's this really cool mnemonic. 16 is 61, and 28 is 82. You don't have to do that 9/5 minus 32, whatever.
For some of you who like Celsius, that would include me, right?
That would include you,
Thank
and
you
all of
very
our
much.
international listeners.
I'm surprised that it isn't warmer here. This is what the weather's like on the eastern coast. Spring weather, kind of chilly. I thought it would be hot. Anyway, our local guest is sitting right next to me, between us. She is a professor here in the Department of Biology, Laura Newcomb.
Hi.
Welcome to TWiV.
Thank you.
And thanks for inviting us. Laura approached me over a year ago, right?
Yes, at ASM, and
She said,
thank you for coming.
I want to bring TWiV to Cal State San Bernardino, which I thought was Santa Barbara for a while. I said, wow, I'd love to go to Santa Barbara.
Sorry.
It's okay. I'm happy to be here. So thanks for having us. So we're going to do a TWiV. We have an audience here. I'm amazed you guys all showed up. Thanks for coming. Is your lunch good? That'd be good? Okay. Try the veal. We're here every day.
How many of you have ever listened to a TWiV before? Raise your hands.
Nice. The ones in the front here. See that?
Quite a large number.
The ones in the back are, you're just here for the food, right? I hope it's worth it. Anyway, we're going to do a real TWiV, which means we're going to do a paper today, an email and picks of the week and all that. But I want to first find out a little bit about our co-host, who has brought this great collection of viruses. Don't let me forget to go through those before.
Okay.
Where are you from, Laura?
Originally, I'm from Milwaukee, Wisconsin.
Born and raised, yeah. Anybody?
Born and raised.
Yeah. Milwaukee, yeah. Cool. I bet you didn't know that, right? Yeah. Went to high school there?
I did, yes.
And where'd you go to college?
I went to college at UW Milwaukee, go Panthers, for my undergrad. And there I worked on methanotrophic bacteria. And then I did.
So you went to college with the idea of being a bio major, right?
I did, yes. I knew right away I wanted to be a bio major.
You didn't want to go to medical school.
I did not want to go to medical
All
school.
right.
No, I wanted to do
Right.
research.
And where did that come from? Do you have any clue?
Where that desire came from? So actually in high school I learned about recombinant DNA techniques, and I thought I had originally wanted to be a veterinarian, but once I learned that I could play around and manipulate DNA, that changed everything. And then I wanted to be a researcher.
Great. So you went to UW, University of Wisconsin. UW is somewhere else, right?
Right, that's Washington.
And then you decided right away to get a PhD?
Yep, so after I spent a long time as an undergraduate, actually. I had a daughter and was a single mom through a lot of that, and spent a total of about 5 years getting my degree, 6 years on campus, taking some time off during that period. So I had a lot of undergraduate research experience, and then went on right away to UW-Madison for my graduate work, which I did not work on viruses there either. I worked on cell cycle regulation in Saccharomyces cerevisiae, and did basically gene expression, regulation of gene expression work there. But I knew I wanted to get into virology from a very early stage. So I've always, even as an undergrad, found viruses very, very interesting. And so after my PhD work, I then went to the lab of Dr. Robert Krug to work on influenza virus.
In Austin, Texas,
In
right?
Austin, Texas, right.
He used to be in New York City.
Yes, but I was a postdoc after he had already moved.
Actually, he was New York, and then he was at Rutgers. For many years, and then moved to Texas. Well, Bob was on TWiV as well, so we now have a complete succession of the Krug lab. We have you as well. And then after that, I presume you worked on influenza virus in his laboratory, right?
Yes, yeah. And that's what I took with me here.
You came directly here from your postdoc? Right?
Yes, yeah.
And what are you doing here? What are you working on?
So we work on influenza virus here, primarily the activities of the internal proteins, so the RNA-dependent RNA polymerase, and along with the nucleoprotein. And we kind of have a focus on the nucleoprotein and its roles in potentially regulating gene expression.
Okay. Now, you probably know I did my PhD on influenza virus, so all kinds of flu work is dear to my heart. Even though I went away from membranes and negative stranded viruses, I still think that influenza viruses are cool. Who's here? I know there's one person from your lab here, right? Anyone else?
Right there.
Well, up in the front. Cool. All right. Anything else, Kathy? Did I forget something? Oh, the viruses. You didn't remind me to. So right in front of us here, we have the coolest collection of homemade viruses, of metal and paper, pipe cleaners, wood, I guess, looks like plastic. What are these from?
Origami.
You brought these, Laura. Where do they come
So
from?
these are actually made by my students from my virology class. One of the first assignments I give them is to make an icosahedral capsid. And I just tell them they need to do something a little bit unique, since viruses are very unique. And so this is what they've come up with.
Which is your favorite? I guess you can't.
Oh, I can't say my favorite. I have a lot of favorites. I do really like the biowarfare one.
That's the green one. That,
the green one.
With the face, yeah. Which the young man over here made, right?
Correct.
Thank you.
I like the one that's lit up that my student made.
That is great.
Alicia.
That is
the
great.
origami is something I can't do, so I find that kind of fascinating.
Right.
And of course, all the bacteriophage are kind of my favorite because I have that artwork on my body.
You have a phage on your body? Really?
I do, yes.
Where? Can we see it?
Sure. So here, hold that. So.
Nice. Look at that. You guys know she had 2 virus tattoos. Did you know this? You shouldn't know it.
You know now.
It's a good thing they're on her arm, right? It could be, you might not be able to
This
show
just
us.
shows my dedication. That phage tattoo, I was only about 21. That was my first tattoo I ever got.
So you were 21 when you got that. Which
Didn't
is,
even study viruses yet. I just
and you
really
just
liked
thought they were
them.
cool.
And I knew eventually I would get to a virology lab.
So when you went to the tattoo parlor, did the person know what a phage was, or did you bring a picture of it?
Actually, I didn't go to a tattoo parlor. A friend of mine did it.
It's probably smart, yeah. So the phage
It
is on
was
the
all
bottom.
sterile, though.
You have a phage, and on the top look like an envelope virus.
So
right?
the top is a depiction of an influenza. It's a little circular.
It's nice.
So, but yeah, and that one has a little color.
I think the nucleoprotein isn't quite right, though.
You can't see the internal proteins.
That's great. I think that's our first virus tattoo on TWiV.
I don't think we've had a virus tattoo before, so
The
this
changing
is
face
really
of the
special.
professorate.
Certainly nobody has, certainly no one has told us about it.
Back to the virus models,
Yeah.
I want to point out that one of them has Mickey Mouse paper on it, so I think that means it's a mouse virus. And since I
agree.
work on a mouse virus, that's near and dear to me.
It can't be adeno, though, right? Doesn't have the fibers,
Doesn't have spikes.
but it could be
The
some other
phage is
virus.
really cool. That's the first one that Vincent and I got to see, the wire phage.
That was
They're
one
all
actually from a student from one of my earlier classes.
Yeah,
I've
they're
kept
just
that one.
fabulous.
These are great. I love it. And I think having a class make this sort of thing is a great idea. I'll have to try it one of these days for extra credit. Is that what you do? Or
Actually, it's the first assignment I
thing?
give them after we talk about structure, and I did steal it from my professor from UW-Madison, Bill McLean, who had us make just the paper virion, and I told them they need to do something a little unique, and this is what it's grown into.
It's beautiful. Nice collection. I have some on my desk, but not as extensive as this. Someone brought me an origami virus, same color, actually. So a student get inspired. And there's a tiny one there, which I really like. There's one here, a red icosahedral capsid with a plastic shell, which is held together by clips.
That's
What do
the
you call-
envelope.
The clips of the glycoproteins,
Yep,
is that
yeah,
right? That's
exactly.
great. That's creative. And then we have this pipe cleaner one. These are great. You guys should look at these afterwards. These are just absolutely great. Thanks for sharing those. All right, we have a paper today for your listening enjoyment. You know, when we're on the road, we often don't do papers. I did it a long time ago in Philadelphia, which was the first live TWiV we did in front of an audience. We actually did a paper during that. I even remember the paper. It was the effect of cytomegalovirus on blood pressure. And there was one person in the audience. It was a room this big, and there was one person. But we did it anyway, because it doesn't matter. This is a podcast. It's recorded. It's sent out there. And those of you who don't know about TWiV, by the way, we have tens of thousands of listeners, steady listeners. You'll see from our email, we have dedicated people who love this because we bring science to people in a different way. Anyway, the paper is published in PLOS Pathogens.
And PLOS Pathogens is an open access journal, which means that anybody can get it. You don't have to have library access.
Right.
So if you want to go look at this or some of the pretty pictures in it later, you can easily do that from any computer.
So I picked this paper for a number of reasons. One, Laura said, I like RNA viruses, so this has to do with RNA viruses. And I wanted to pick something that would appeal to the class here and the students and so forth. And this has to do with mosquitoes and viruses. The title is A Neuron-Specific Antiviral Mechanism Prevents Lethal Flaviviral Infection of Mosquitoes. First author is Xiaoping Xiao, and the last author is Gong Cheng. This is from Beijing, People's Republic of China, Tsinghua University, National Institute for Viral Disease Control and Prevention, also in Beijing, and Department of Microbiology and Immunology at Medical College in Valhalla, New York, which is not too far from me. And so this is all about the fact that mosquitoes, as you know, transmit a number of virus infections among humans and among animals. But the mosquitoes are pretty much okay with this. They don't get sick. Viruses like dengue and West Nile, Japanese encephalitis virus, yellow fever virus, they replicate in the mosquito, and that's mostly a requirement for transmission to amplify. And of course, the salivary gland has to be a site of replication so the mosquito can inject it into the host. And of course, this happens when the mosquito takes a blood meal. And did you know that only female mosquitoes take blood meals? So they are the ones that transmit virus infections. The guys are just, sleeping in the woods somewhere, probably. So the virus has to replicate, but the mosquitoes are fine. And that's what this paper is all about. Now, in contrast, when these viruses get into people, they cause problems. They cause neurological problems. There's significant disease and significant death associated with these infections. So the question is, how do the mosquitoes control the infection? And this is a kind of a pervasive question in virology. Bats are similar. Bats are full of viruses, but they're okay. We're actually also full of viruses, and we're okay most of the time. So I guess it's a universal thing. But people are really interested in mosquitoes and bats because they're vectors for human infection. So the focus in this paper is a protein originally discovered in Drosophila. It's a protein required for neural development in the CNS. It's called Hikaru Genki. Anybody speak Japanese here? Hikaru Genki. I don't know what it means, but it's pretty cool. And the name of the gene is an abbreviation of that, Hig, H-I-G. And this is an interesting protein because it's made up of modular components. It has an Ig-like domain, immunoglobulin-like domain, which is very common. Poliovirus receptor has an Ig-like domain. And it has 5 complement control protein domains. And these are also called sushi domains. So eat sushi, the bottom line is eat sushi and you will be protected. Now that's not true at all, but you can eat sushi because it's great. Sushi domains are repeats in proteins. They're about 60 amino acids long with a number of conserved cysteines and tryptophans. And they're found in the proteins of the complement pathway. And you know this is a series of blood proteins in mammals that protect against microbial infections. There are lots of protein-protein interactions that happen in the complement pathway, and those interactions are mediated by these sushi domains. All right, so this protein in the mosquito, in Drosophila Hig, has sushi domains. So they found these sushi domains, by the way, not only are good for binding other proteins of the complement pathway, but they apparently can bind viral proteins. So some of them are receptors for viruses. The Epstein-Barr virus receptor, the measles virus receptor, these are sushi domain-containing proteins. And then there's some other proteins that are involved in recognizing viruses as foreign. So they have an ability to recognize virus particles. And that's going to be an important part of this story. So this paper concerns Aedes aegypti, right, which is the vector for dengue and yellow fever. West Nile and Japanese encephalitis viruses have also been isolated from this mosquito, but it's not the vector for this virus to be transmitted. So they find a homologue of this Hig protein. Remember, it's in Drosophila. They find it in Aedes aegypti. And they look phylogenetically, and they find this protein is conserved in many different insects. And here includes Diptera, Coleoptera, Hymenoptera, Lepidoptera. Now, if Dixon were here, he could tell us.
I can tell you some of those.
The Diptera are flies.
That's
right?
flies, right?
Coleoptera?
Moths, maybe? Is that right?
What's a Coleoptera? Anyone know? All right, you can. If I had internet, I could look it up. But usually when we do this, we're on Skype and we're Googling. Hymenoptera? Hymenoptera? We're going to get email.
Yeah, we are.
I know Lepidoptera are butterflies, right?
Yes.
Is that right? Okay. Anyway, it's in a lot of different insects, but not all of them, and it's not in vertebrates. This is the cool thing. This Hig protein with the sushi domains, we don't have it. Vertebrates don't seem to have it. And I'll get back to that because I think that's really, really interesting. So the first question they ask is, where is it in the mosquito? So they cut up mosquitoes and they look at RNA, and they also raised an antibody against the protein. They produce the protein in, I think, insect cells, make an antibody. And it's mainly in the head. And the head, of course, is mostly brain. And so it seems to be a neural-specific protein, or almost neural-specific, just like it is in Drosophila.
They also find it in the carcass. Does that just mean the exoskeleton?
I think it's the whole thing except the head, right? So they say that they think that's probably peripheral nerves that must have this protein in it, right?
I didn't get that detail.
But they look at ovary, hemolymph, which of course is the circulatory fluid, midgut, salivary glands, pretty low. But the head has a lot. So this appears to be, and they actually look in a bit by in situ to see that it's specifically in the brain. The protein is, if you take a plasmid encoding this protein and put it into cells, the protein is secreted, but it remains on the plasma membrane. So somehow it gets attached to the plasma membrane. It doesn't go off free. And so this is important because this is going to play into the function. They have a nice immunofluorescence where they stain cells, and they use wheat germ agglutinin to show this is the plasma membrane, and that's where this protein is localized. So again, we're talking about Hig, what is it again? Hikarogenki, Hig in mosquitoes. They do some sections of the head of the mosquito and stain with antibodies to the protein. You can see exactly where it is. It's in the brain.
It's in very specific parts of the brain, in the optic lobe and the antennal lobe.
Right. Okay, so it's in mosquitoes, it's in the brain in specific places. What is it doing there? So they want to know what happens if you got rid of it and then you infect the mosquitoes with virus. So they've got their antibody, and they inject the virus into mosquitoes. I think intrathoracically is the way they inject it, right? So it goes right into the body cavity, and then it gets into the circulatory fluid. So they mix virus, dengue virus, with an antibody to this Hig protein and inject it, and then they look in the whole mosquito at dengue virus RNA after 3 days and 6 days. And basically, when you inject an antibody to Hig, you get more virus RNA. So taking Hig away apparently is removing some kind of defense against dengue virus replication. Now, these are all done by PCR, basically. There's no plaque assays, which is unfortunate, because I love plaque assays.
Right.
I mean, but more than I loving them, they represent infectious virus, right? Do you do
Right.
plaque assays in your lab? I'm
Yes.
sure you do, right?
Yes.
So measuring RNA, actually in some of these cases, the difference between control serum and the anti-Hig serum is very minimal. There's not a huge effect. In other cases, it's big. And I just wonder if they looked at infectious virus, if they would see more of an effect of this. Of course, infectious virus is what's replicating, where the RNA, you don't know if there's an excess of pieces of RNA or something. It's something I always come back to. I know that they can do plaque assays with dengue, so. They have lots of supplementary data in this paper. I mean, the actual paper is huge, right?
The paper itself is huge, and then the supplement is, you said, I thought it was 15 or 19 supplemental figures.
Right.
Most of the things they do in this paper, they do with dengue virus, and then they also do them with Japanese encephalitis virus, this
Right.
related virus. So a lot of the Japanese encephalitis virus data ends up in the supplemental material. So it probably could have been 2 or 3 papers, but they wanted to tell this one story, so it's huge.
It's a huge amount of work. So Japanese encephalitis is another neurotropic human virus transmitted by Aedes aegypti. They show that basically if you remove this Hig protein by giving the mosquitoes antibodies to Hig, at the time that you give them virus, you get more virus replication in the whole virus and also in the head. And here the differences are more than for dengue. You get a much greater effect with JEV. They did the same experiment in Culex mosquitoes, which is the natural transmitting species of Japanese encephalitis virus. They cloned the Culex Hig cDNA, and they did similar experiments. They actually do, and I think I skipped this for the dengue, they do double-stranded RNA knockdown. So the cool thing about insects is that you can put double-stranded RNA into them, into whole insects or into cells. They take it up, and that silences the target. It's really very efficient, much more so than mammalian cells. Those of you who have tried siRNA knockdowns should all switch to insect cells because it's really very efficient to do that. So they knock down the Hig in the Culex species, and again, it affects viral loads. The loads go up when you remove the Hig, either by knocking down with double-stranded RNA or by antibodies as well. And by the way, you can actually feed these mosquitoes a mixture of virus and antibody to this Hig protein. You can feed it to them, they will get infected, and the antibody will still reduce the endogenous protein. Remember, Hig is mostly in the CNS, right? So that means after feeding, this antibody is getting into the brain of the mosquito. That's pretty neat that's happening. And there it can apparently block the protein, and it abrogates this function. And again, it's not eliminating virus replication, but apparently it keeps it at a low enough level so that the mosquito's okay, right? That's the key.
And it's mostly inhibiting it in the brain, because that's where
It's mostly in the brain,
expressed.
exactly. So that's where, exactly. So it's a good point. These viruses, dengue and Japanese encephalitis, replicate in multiple tissues of the mosquito, but this protein is just in the brain, and that is where it's kept down. They do some very nice experiments where they show that in a normal mosquito without any antibody you infect with dengue, you don't get a lot of apoptosis in the brain. But when you add antibody to Hig, you get an increase in the amount of apoptosis, which you can see beautifully in these sections where they're using caspase antibodies or tunnel assays to look at apoptosis. So then what does that do if you get apoptosis in the brain? Well, the mosquitoes die, right? And they do a survival assay as well, and they show that if you give mosquitoes virus plus an antibody to this Hig protein, their survival goes way down. Now, this is interesting. I found that they have a survival curve from 0 to 25 days. So they inject their control mosquitoes with pre-immune serum only, and about 20 to 40% of them die over the course of the experiment. So that's the natural attrition rate, I guess. Because they say, and even with dengue, it's the same, you have the same attrition rate, because they say dengue doesn't make the mosquitoes die any more frequently than if you didn't infect them. And so apparently raising mosquitoes in the lab, or maybe in nature as well, they die naturally quickly.
Yeah, I always thought insects didn't have a very long lifespan.
How long do mosquitoes live? Do you have any idea?
I don't know.
Again, Dixon should be here to tell us, or Google it, but we can't Google.
It's online.
So now you see, revealed is the extent of our ignorance, right? Without Google. So if you knock
Jim Ferrari here? Anyone from Jim's lab? Okay, he would know too.
You guys can yell things out. We don't mind. This is okay for you to do that. Okay. Anyway, I bet mosquitoes don't live more than a month or two. right? So maybe this is the natural attrition rate. The point is that when you add antibody to Hig, the mosquito, they're all dead within 25 days, whereas, just 40% are dead within the same length of time with the control, which is pre-immune serum. Okay, so now how is this working? It's quite clear that this protein is somehow lowering virus levels in the mosquito so that apparently they can live with it. Because if you take away the protein, the virus replicates to high levels and the mosquitoes die. So how's it working? It turns out that Hig, they can make the protein, of course, in cells. It binds viruses. It binds virus particles. So they have a variety of pull-down assays with their antibody to Hig. They have tagged versions, and they can show that Hig interacts with dengue virus particles. You can bring them down. It also interacts with the viral glycoprotein in the envelope. All right, so this shows that Hig is somehow directly interacting with the virus. And they also look in mosquitoes to see if this is the case, to see if Hig and the virus co-localizes. And they actually have some very nice staining experiments using sections of brain again, showing that the Hig protein determined by an antibody is co-localizing with the virus. So they're right there at the brain cell surface. They go on to map exactly what parts of the viral glycoprotein interact with Hig. So they do a number of really nice experiments where they produce different lengths of the protein. This Hig protein, of course, has both the Hig protein and the viral glycoprotein. They do this with on both sides to map the interaction. And they can show that the interaction on both sides is in a very specific region. And in fact, they can produce the protein, the different truncation versions of the protein, and also inject them into mosquitoes and show that the full-length ones are protective, but the reduced ones are not. So it turns out for Hig, you need the second to the last complement control protein to get protection against infection. So again, this has 5 complement control proteins. They make a series of deletions to get at that. I think they've also done this with another virus as well. So the glycoprotein of dengue, the E glycoprotein, which is in the membrane. Do you have a dengue here, by the way? None of these are dengue, right? Oh, those are people's names on them. Look at
Right.
not virus names.
Well, the red one with the envelope could be a dengue kind of.
That could be a dengue. So the glycoprotein in the membrane is what is binding this Hig protein, and a very specific part of it that they can show by these deletion experiments. All right.
Are you ready for the digression
Yeah, go
about
ahead.
the lifespan of mosquitoes?
You got
Okay,
it?
so Wikipedia first told me that the lifespan of a male is 0.027 years. So then I had to go to my calculator
27
and
years.
figure out that that's 9 days. So then I had to go back and find out that the lifespan of females can be up to 6 to 8 weeks.
Okay.
So presumably they're doing these experiments on females because the males would be gone.
And the females are the ones that
The females are the
transmit.
ones that transmit, so we want to know. Although I'm sure male mosquitoes have this protein as well, but it doesn't matter because they don't transmit infection. I don't even know if male mosquitoes are ever, it's a good question, are ever infected with
I
viruses.
was going to
right?
ask you that if
I don't know. How would a male mosquito acquire one of these viruses? I don't know if they're sexually transmitted,
Yeah,
right?
if it's vertical transmission maybe. Yeah, I don't know.
Could be.
Again, we'll get mail.
We have
Someone
people
will
who know
tell you.
these things. Anyway, so one of the interesting parts of this series of experiments is they have a truncated version of Hig-E, lacks the last 2 complement control domains. It doesn't block infection, but it can still bind the virus. So somehow that second complement control domain is probably interacting with something else, and that's what's blocking virus infection. Let's see, what else? Lots of experiments here. Okay, the last experiment, how does this binding to dengue virus reduce virus replication? So that's really the question in the end. They thought of a lot of ways that this protein might induce innate pathways like RNAi, antimicrobial peptides, these are all known to be operative in insects, reactive oxygen species, melanization, the induction of this melanin. But they looked at all these pathways. There's no change when they take away Hig. So then they started looking at, because Hig binds virus, they said, well, does it affect virus binding? Does it affect virus entry, right? Makes sense. So they mix virus with or without Hig, and they add it to cells. There's no effect on binding. It's interesting because Hig is binding the virus particle, but the virus can still bind to cells. So it's apparently binding somewhere other than the site on the glycoprotein that interacts with receptors, right? And then they show that the uptake of the virus into cells is inhibited by the Hig protein. They have a marker for endocytosis. There's a protein, a Rab GTPase, that Rab5 is a GTPase that's a marker for endocytosis. And they show that dengue normally goes into the endocytic pathway, but when you add Hig, the virus just stays on the cell surface. So you may ask, is it a general inhibitor of endocytosis? And they actually address that. It's not, because they can look at the uptake of beads. They have these fluorescent beads, by the way, that get taken up by the endocytic pathway. They're nanobeads. And then you can measure endocytosis by flow cytometry. This is brilliant, right? And they show that Hig does not prevent endocytosis, but it's blocking specifically dengue uptake, right?
I just wanted to describe that when they do those binding experiments and the entry experiments, they're almost the same experiment, but the binding experiments are done at 4 degrees Celsius, and then you do the entry experiments by raising the temperature, and at a higher temperature, I think they use 28 or 30 depending on the cell line, then that's what's going to allow the virus to go in, if it's going to go in, but it's blocked in the case of entry, but it's still able to bind at the 4 degrees. So those are standard kinds of assays that people use for binding and entry of viruses.
So it's kind of emphasizing that they can be 2 different steps, the binding versus the fusion and entry. Right. Yeah.
The interesting thing here is that they do these experiments with Synbis, which is an arthropod transmitted virus, but it's different. It's a alpha virus genus, right? And it is not inhibited by Hig in insect cells. So this appears to be a Flavivirus specific mechanism, which is quite interesting. Again, Flavi's West Nile, Japanese encephalitis, dengue, yellow fever, etc. And the other part, which I think is really interesting, is so remember, vertebrates don't have the Hig protein. So they take a human cell line and they put a plasmid for Hig in it, make the protein. It does not block Flavivirus uptake in human cells. I thought that was really interesting, because that means there's a whole machinery probably in insects that's doing this, not just Hig, right? And that's going to be really cool for them to look at. I mean, I guess you just start looking at interactors of Hig protein, right? That's what I would do. So this is cool. This is a brain-specific mosquito protein that, not just mosquitoes, but other insects as well, that can prevent uptake of a number of flaviviruses, and it keeps the virus levels low in the mosquitoes so the mosquitoes are healthy, all right? And they say this may have been selected during evolution to let mosquitoes live long enough so that they can transmit infection. Now, I was thinking about this a lot because in humans, and I think the same is in mosquitoes, once a virus gets into the brain, the central nervous system, it typically doesn't get out again. So what would be the selective pressure to let a mosquito live longer? And I guess it's replication in other tissues. If you don't live long enough, the virus can't.
Right. I think they allude to that in discussion, that allows them to then be able to transmit the virus onward if the mosquitoes survive.
Right, because if they don't survive, they can't transmit it from the salivary glands, obviously. So it's kind of an indirect selection. Right. But what I'm wondering is why we don't have Hig proteins. You know, because we get CNS infections and it's problematic. Why haven't we evolved to have, and you know, all of our ancestors, the primate lineage and so forth, there's no Hig proteins. Why not? We have lots of neurotropic virus infections.
You live longer than 9 days.
How do you know? I live long. Yes, humans live a long time, but obviously other selection pressures come to bear on human evolution and select for certain genes. So it's not that it can't be done, but there must not be. I think we transmit infections well enough that this is not a consideration, I suppose. It's not our mosquitoes, it's what they do for a living, I guess. They transmit infections.
Maybe there's some mechanism that just hasn't yet been discovered. I mean, it could relate to the fact that bats have all these viruses and don't seem to be affected by many of them.
So that's the other thing that I think is interesting. I wonder, bats don't have this protein, but maybe they have similar mechanisms. And people, as you know, we've talked about this on TWiV, people are trying to figure out what makes bats resistant to viruses. A lot harder to study than mosquitoes, though. But I think this is a pretty cool area. There's a little bit of work already done on this as well, but it's not for everyone because you need to be able to grow mosquitoes and infect them. But they say Aedes aegypti are nice to grow in the lab and can infect them and so forth.
Something I was interested in is, could this be, they talk a little bit in the discussion about trying to target the host vector, right, so that we don't get West Nile virus infections, right? So what if we could get this protein expressed in the cell, in different cells to keep the virus not producing so much in the salivary glands? No, in the host, in the vector
in the mosquito.
Host. Yeah, somehow treat them. I don't know if that would be possible.
So it would kill the mosquitoes and they wouldn't be able to transmit as effectively?
No, not so that it would kill them. Don't inhibit the protein. What if we could get it expressed in other parts of the mosquito?
Oh, like the salivary gland.
So get rid of it completely.
Oh, yeah,
Get rid of the ability of the host
That'd be great.
Carry the vector.
Yeah, so you make a transgenic mosquito, and in theory, it shouldn't transmit if you can target this to the salivary gland, right?
Correct.
Yeah.
That was my
Of course,
question.
the problem with that is just get those to propagate in the wild, right?
Yeah, exactly.
Because to have them,
You'd have to have some spray you could.
somehow you have to select for those
Influence.
mosquitoes. Anyway, I thought this was pretty neat. I hope you guys liked it.
One of the things I wanted to point out is that insects, or at least mosquitoes or Drosophila, have the equivalent, sort of the equivalent of the blood-brain barrier. They have septate junctions that protect their brain from getting infected. There are only certain things that can pass in the same way that we have a blood-brain barrier. So they have multiple ways to try and protect, and they have this additional Hig that, as Vincent pointed out, doesn't seem like humans have.
All right, that's our paper, and hope you learned something from it. I did. I think it was cool. And now we have some email. I wonder if you could start with that first one, Kathy.
You have to wait while I get that up. But meanwhile, so you'll get to read the first one. But first I wanted to say that we're here as part of the 4th Annual Student Research Symposium, Meeting of the Minds. And this is the program for it. It's really been interesting. This morning we heard a couple of research talks. And then while Vincent was setting up, I got to see a couple of scenes from the opera that was presented 2 weeks ago here, and it was fabulous. It was in English with lots of Spanish words, and the husband kept correcting his wife's Spanish, and it was really well done. I wish I could have seen the whole performance, but it's a really interesting day. And then after this, there's going to be posters and some concurrent student talks and things. And then Vincent has another lecture. This isn't a lecture. Vincent has a lecture later today where he talks about communicating science. So it's full day of activities here.
Yeah, thanks for having us.
Thank you
Appreciate
for
that.
coming.
Do you want me to take that first one, Kathy?
Sure, because I'm not
All
there
right,
yet.
This is from Benji. This actually is sort of a follow-up to, I think, 2 episodes ago. Dear Vincent and friends, I'm a 3rd-year medical student at University College London, doing some research in oncolytic measles virus, and enjoy listening to your show regularly. I couldn't help but pick up on the question from yesterday about why IRF7 deficiency should cause leukemia, and what on earth interferon has to do with anti-cancer immune surveillance. So just to explain this, we did a paper a couple weeks ago where a child that had developed severe influenza was found to have mutations in the gene for IRF7, an innate immune gene. And in the paper they say that this is associated with leukemia. So why would innate immune deficiencies be associated with leukemia? And so Benji continues, well, for my field, I can confirm that a whole series of studies, which he provides references for, have shown defective type 1 interferon responses in cancer cells, such as ovarian cancer cells. And in fact, that's why you can use, in part, viruses for oncolytic therapy, because tumor cells are typically deficient in innate responses, and the viruses replicate really well in the tumor cells. But why would a cancer cell activate interferon pathway in the first place? I haven't been able to find an explanation in the literature, but I have a hypochondriac's cancer theory that I'd love to share with you. During the rapid cell cycling of a cancer cell, they may well have lots of cytoplasmic DNA and RNA floating around while they rapidly dive in and out of mitosis. But RNA and DNA floating around in the cytoplasm looks like something else to the cell, a virus. So some cancer cells could be getting tricked into thinking they are virally infected and start producing lots of interferon, stop translation, activate NK and T cells nearby. Some successful cancers, though, will have a defective or inhibited component or two of the interferon pathway and survive. So this could be a tidy explanation as to why patients like patient P with defective IRF genes are more likely to develop leukemia. Their cells are just as hypochondriac as everyone else's, but they don't have the phone number, interferons, to call the doctor. You like that little analogy? I think that's great. What do you guys think? I've made an assumption or two along the way, but I can't for the life of me think how to prove that there's RNA and DNA hanging out in the cytoplasm of cancer cells. Do you have any advice for how to measure that? So the idea is that these cells are having lots of excess RNA and DNA in the cytoplasm as a consequence of being transformed. I'm not sure that that is necessarily true, but you could do in situ looking, but RNA is in the cytoplasm anyway, so I think that'd be really hard to quantify. I just don't know. I've asked a number of people why tumor cells are deficient in interferon pathways, and I'm getting blanks, so I don't really know the answer. Does anyone know? Why would a tumor cell be deficient in interferon? It must have something to do with surveillance. Somehow interferon pathways involved in early tumor surveillance that we're missing, and that's why they can escape it, but I just don't know. So if anyone out there knows. All right, you want to take the next one, Kathy?
Okay, so Timothy writes, Dear TWiV family, thank you again for showcasing my work on episode 273, Lambda is not just a phage. A non-virologist crystallographer friend of mine at Rockefeller sent me a note after that aired saying that you know you've made it when you are on TWiV. It's really an honor and yet more confirmation that the breadth of your audience is wide. I've been experimenting with new ways for me to share scientific information. Since the previous version of that LCM movie I'd made didn't have a voiceover or much context, I decided to embed the LCM video with a video abstract. I talk about the techniques we use in our cell host and microbe paper, and I talk about results and context in broad strokes. See the link below. I hope you enjoy it. I hope that this movie and future movies will help increase public engagement in virology. Keep up the good work, Tim. So he includes a link to a YouTube, and I want to insert at this point that all of the questions that we read on each week's TWiV get posted on the web page, and so you can go to those questions and you could find these links, and so you could check out this YouTube where this person has now added the voiceover to his LCM movie. It's pretty neat.
So LCM is laser capture microscopy, right? I think it's
laser capture microdissection.
Microdissection. It's a way of getting very specific parts of a tissue. So he did this with infected livers, if I remember. He'd cut out very specific portions and then look at the sequence. It's just fabulous, fabulous. You don't have the show notes, so you can't read any.
I don't.
Right?
I can give her my iPad.
No problem.
It's okay.
All right, the next one's from Richard. Dear Twixonauts, that's a new one. I'm a registered nurse in Illinois, so you get a sense of who's listening, all kinds of people, and very much appreciate your podcast. I ran across a fictional application of parasitism in a science fiction novel entitled Blind Lake by Robert Charles Wilson. It's not a spoiler to describe it on your episode, as the particular biological mechanism in question does not relate directly to the plot. The novel describes an alien species with a technological civilization that is diurnal, working by day and sleeping at night. Every night, small creatures enter the home to suckle a body fluid secreted by small nipples for the purpose. When an individual adult dies or becomes disabled due to injury, illness, or old age, the desire for a replacement causes the members of its group to activate an endogenous virus. The virus then infects the parasites, some of which begin a process of maturation, culminating in the development of adult aliens. The author does not elaborate further, but it occurs to me that this reproductive modality allows for mechanisms for genetic recombination distinctly different from that seen in sexual reproduction. Individual parasites might be co-infected by viruses produced by more than one adult. So if the virus is segmented like influenza, the virus might recombine, or I would say reassort, right?
I would say reassort, yeah.
Furthermore, if there were transmission between individuals at the parasitic stage, the virus could carry genes from one adult to another. For those listeners who prefer to skip to this description, it may be found on the 7th page of chapter 33, though I imagine that the author would prefer that one purchase and read the entirety of his book. I haven't heard of this one. Has anyone read Blind Lake? No? All right. You did?
Yeah.
It's good. That's not a good sign. You don't remember it. Okay, I might check it out. It's an interesting thesis. But authors like to come up with these convoluted scenarios. I have a few authors who have contacted me and they want me to check over their ideas for books. And I always tell them, this doesn't happen in the real world. And they say, well, I'm not writing about the real world. Yeah.
So Greg Bear wrote several books. I don't know if you've read
Yes, yes.
Greg Banner.
Darwin's Radio.
Right.
And
Darwin's Radio.
Children, both of which have endogenous retrovirus activation as
Right.
a major problem.
Greg Bear, yes. Endogenous retroviruses fascinate authors. So this one and a few people who have contacted me, I don't know, maybe because they're in our genome and the idea is they can do different things, right? It's pretty cool. Here in Chicago, it's sunny, 50 degrees Fahrenheit, 10°C, wind 8 miles per hour, dew point 26. The neutrino flux is 65 billion particles per square centimeter per second. That's the new one, isn't it?
We have never had the neutrino flux before.
All right, you're next, Kathy.
Okay, Erica writes, to the fabulous TWiV team, I wanted to say thank you for making this amazing podcast. I found TWiV a little over a year ago while looking through iTunes, and it has been a fantastic weekly addition to my life ever since. I graduated in 2014 from the University of Florida with my bachelor's in microbiology and cell science, and I'm currently working as a laboratory technician at the university. It's hard to believe that this person just found TWiV and they've been at Florida all this time. One of our co-hosts is Rich Condit, and he's a professor at the University of Florida. So anyway, working as a laboratory technician at the university. I knew I wanted to work in virology before TWiV, but this podcast has helped solidify and intensify my dream of becoming a virologist. After listening to the podcasts and perusing the website, I've begun thinking about doing science communication work in the future. I want to work to communicate topics in science in a simple way to the general public to attempt to bridge the gap in science literacy. I don't think I would have ever realized this passion without the ideas from TWiV. My biggest thank you is for the advertisement for the Icahn School of Medicine at Mount Sinai's PhD program. I applied after listening to the advertisement on TWiV and interviewed this January. I fell in love with the program, faculty, and of course, New York City. I'm beginning my PhD with them in August, and I don't think I would have ended up with this amazing opportunity without TWiV. I recommend this podcast to everyone I know, whether they have a science background or not. I just want to say thank you for the long-lasting impact you've made on my life, and keep up the amazing work. P.S. We are having beautiful weather here in Gainesville. It's 80 degrees Fahrenheit, 20 degrees Celsius, partly cloudy with 59% humidity, and winds at 9 miles per hour, 14 kilometers per hour.
What a great story. By the way, you can advertise your graduate program on TWiV. It's very reasonable, and you get results, as you see. It's a focused audience. It's people who are interested in science.
It also helped that they were advertising in the fall when we had about 6 episodes in a row that were solely devoted to Ebola.
Right.
And the Ebola in the title names brought in, I think, a lot of additional listeners.
Yep. Yeah, you can't always count on that. But we still, I think we have a good reach for this audience. Well, that's great. You're going to have to visit the TWiV studio, Erica, sometime. Check it out. We have one more here from May, who writes, Dear Vincent and the TWiV Masters, love the show. I came to TWiV via Vincent's virology course on YouTube. Since I'm a sales rep for a biotech company, I have tons of windshield time. I've never heard of that one before. Think of driving from Johns Hopkins to Virginia Tech. I sustain on audiobooks and podcasts. Ever since I discovered TWiV, the only voice in my car is from you guys. I love the show so much that I downloaded the Microbe World app so I can start from episode number 1 today. Just finished episode 100. Today I just finished episode 100 where you interviewed David Baltimore. He was at Johns Hopkins a month ago. I could not make it to his seminar. I wish I could. Bought the book Ahead of the Curve. You recommend it on Amazon. I learned a lot from the show. For example, I learned from TWiV that CMV is the only virus that can cross the placenta. I think I sort of know. Sadly, a friend of mine had CMV when she was pregnant, and her baby girl had lots of problems from the infection. But nobody told me point blank CMV is the only virus that can cross the placenta. TWiV clarifies or solidifies that idea for me. So I walk into a lab today before listening to the Baltimore episode to follow up on a project. It's a CMV lab, so I asked the PI, have you or anybody in the field figure out why CMV is the only virus that can cross the placenta? What makes this virus so unique? He said, that is what this project is about. We want to tag the virus with a reporter, mutate different genes, and see which one makes a difference. Wow, I just have a new level of understanding of what he's doing. Or is it sales rep? You go from lab to lab, right? And bother you, and you don't want to talk to them. She's talking science because she learned it on TWiV. It's great. This is not the only example. I'm embarrassed to admit that it's from TWiV that I learned RNAi does not quite work in mammalian cells. I don't know how I missed that. I used to call on Tom Tuschl and Andy Fire. I read genetic engineering news, Nature Methods, etc. Huh? I'm also glad to hear that LIPS, luciferase immunoprecipitation system, is catching on. I remember talking to Peter Burbelow, who developed the method 10 years ago, and I was skeptical. I think the CDC person mentioned the assay in an episode about MERS virus. I know a lab at Virginia Tech adopted the assay. It's more sensitive. More labs should try it. Just want to say thank you for the great show you produce. Keep up the good work, and please, please do not stop producing the show. I'm addicted to it. The weather here is hot and muggy, 27°C, 52% humidity. It's a pre-show of August. Regards, Mays in Rockville, Maryland. All right, that brings us to Science Picks of the Week. And I will have you, our guest and host, Laura, tell us what your pick of the week is. First.
Well, it's not really a pick of the week per se, but but I was looking at a paper, you know, I'll say it's because it was of interest to me because it's something I research, so it's near and dear to me, but it kind of talks about also RNA interference. So it was a paper that tries to use small inhibitory RNAs to inhibit viral infection, and in this case they were showing inhibition of influenza virus infection by targeting conserved regions of the nucleoprotein. And the nucleoprotein is what I'm interested in, and also interested in the conserved regions, but mostly protein and amino acid sequence, right? So this was using RNAi, which targets the RNA, right? And so RNAi techniques can be a little iffy, I think, for trying to do viral therapies because of the fact that viruses mutate so rapidly, the genetic code is degenerate, and so I would think there would be escape mutants pretty readily. So I was interested in this paper to see what they had found, and they did identify a small interfering RNA that could inhibit a number of different NPs, even with slight sequence variation, but unfortunately they didn't do an experiment to look to see how rapidly we would get escape mutants against the siRNA, because I think that's probably going to be an issue. The other reason I looked at it is because it was work from Italy, and I'm going to be going to Italy for the negative strand virus meeting, so I was looking at what research was being done there too with influenza.
So the idea is it's a conserved sequence, so it hits a lot of different viral strains, right?
Yes,
right.
yes. So it's highly conserved in many of the different subtypes and had, you know, broad efficacy. But I do worry with these nucleotide targeted therapies. I would think you would need to have multiple different siRNAs targeting multiple genes to try to avoid that escape mutant possibility.
Do you speak Italian?
No, I do not.
Non parli italiano.
I did get an app that I'm going to be focusing on in the next few weeks after I turn my grant in.
Right. Where are you going in Italy?
The meeting's in Siena.
Beautiful.
Nice.
Siena's
I've
lovely.
never been there. I'm really looking
You'll
forward
love
to
it.
eating a lot of pizza.
Il paglia. It's what? No, not pizza. Other things.
Pizza,
Pasta.
Pastas and meats. They have great meats up there in
Good
Tuscany.
wine.
It's wonderful. Yes. Tuscany.
Gelato.
Yeah. All right. Have fun.
Thank you.
Kathy, what do you have?
I picked something that's called time-lapse mining from internet photos, and it's weird when you first click on the link. It's kind of a link to a internet paper, but they have a PDF at the end of it. The first thing is a video that you can watch. Basically what they do is they mined 86 million photos from the internet that people have posted, I suppose on things like Flickr and so on, and then they put them together for certain sites or glaciers melting or things like that. And they used software that, so they sorted them by date and then a common viewpoint, and then they used software to stabilize the appearance of these, compensate for lighting effects, minimize flicker, and they do some warping things. So this is incredible software that they do. And then you can watch this glacier disappear over time, or changes in a rock monument in Lucerne, Switzerland, or a skyscraper being built in New York City, or they do a seasonal one. They do both Lombard Street and Butchart Gardens in Victoria, Canada. Lombard Street being that zigzaggy street in San Francisco. And so you can see sort of spring, winter, summer, fall to see a change over that kind of time span, whereas some of these other ones are longer days. So I just found it fascinating.
It's cool. Just from assembling pictures out there, you can do all this. It's
Yeah,
amazing,
they
'cause there's
just,
so much.
it's just mining what's out there on the internet.
And it's all made possible by digital photos. You wouldn't be able to do this previously. It's just
Right.
great.
Right.
My pick is a summary of a meeting that took place at the National Academy of Sciences not too long ago. It was called Does the Public Trust Science? So the National Research Council has a roundtable on public interfaces of the life sciences, and they run these meetings about communicating science to the public. This was a 2-day workshop to explore all about trust in science, what the elements of trust are, how trust is built, maintained, or lost. So they had a number of presentations from people who work in science communication. It's a really interesting question because, a lot of people out there don't trust scientists or science, and it's really important for us to work on that. So the videos and the slides from all the talks are available at a website, and I'll put that link in the show notes. So again, that's Does the Public Trust Science? I tweeted this out when it first came out a few weeks ago, and someone immediately tweeted back, do scientists trust the public? Interesting. I didn't answer. We have a couple of listener picks. Paul sent a link to an XKCD page called Degree Off, and I can't look at it to tell you about it in detail because I don't have internet, but it's all about getting a PhD, I believe, right?
How many of you have ever seen XKCD comic strips on the internet?
Yeah.
A few of you. So
You should check it
this is
out.
a good
It's
one.
pretty funny. And then Peter writes, greetings, Vincent, Dixon, Alan, Rich, and Kathy. I have a couple of listener picks. Swedish statistician Hans Rosling discusses how West Africa has managed to contain Ebola virus. Gives a link to that article. There is more from Hans Rosling at gapminder.org, a Swedish nonprofit organization which promotes a fact-based worldview. Some more podcasts that may be of interest. On a recent TWiV, you mentioned the journal eLife. Browsing their site, I see that they have a podcast. The podcast is produced by Dr. Chris Smith and the Cambridge University team that also produces the Naked Scientists podcast. So eLife is a new open access journal. They have a podcast. And I'm a little sad they didn't ask the TWiV team to do their podcast. I don't know why. In fact, one person from eLife came by a few weeks ago, and I said, we do a podcast. You should have picked us.
We don't have a catchy name like the Naked Scientists
The Naked
podcast.
Scientists. It is a good name.
I like TWiV.
Thank you. All right, that'll do it for our picks, and that'll do it for TWiV 338. All the episodes of TWiV, 338 episodes, are all free. You can find them online at twiv.tv, at iTunes, and any app on your phone or mobile device that grabs podcasts can get it and subscribe to it. We do this every week, so you should check it out. And if you have questions or comments, send them to TWiV at twiv.tv. We have a Facebook page, facebook.com/thisweekinvirology, and lots of other things as well. I want to thank everyone for joining me today. Kathy Spindler is from the University of Michigan at Ann Arbor. Thank you, Kathy.
Thanks. I want to point out that Vincent is also has been putting together other podcasts. So if bacteria are more of your thing, there's This Week in Microbiology. There's also This Week in Parasitism. Neither of those is quite weekly from my understanding. They
That's
talk
right.
about what's happening that week, but This Week in Virology is weekly. He's also doing one called Urban Agriculture. You can check out all of those if one of those areas is of more interest to to you than viruses, although I can't imagine why that would be, because viruses are the coolest.
Viruses rule. They're amazing.
Right.
Clearly, I think they're the
So
coolest.
anyway, I want to thank Laura for having me here, and it's been really fun to be here in San Bernardino, so enjoy it.
And I want to thank Laura Newcomb, our local host, who is a professor here at Cal State San Bernardino. Thank you, Laura.
Thank you very much for coming. I'm very happy you made it.
Pleasure. And I'm Vincent Racaniello. You can find all I do at my blog, virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.